We're trying to collect the brain scans from the subject while the subject was watching the movies.
We take brain scans every 2 seconds, and we look at the whole brain to see how the brain activity goes up and down at every place in the brain.
After we acquired the brain scans from the German subject, we used deep learning models to reconstruct what the people see. And what is more interesting is that the model also can predict how the brain interprets what the person sees. For example, when it is a person, then the model can predict it's a person.
What's new in this study is that for the very first time we showed that we can use a convolutional neural network to understand how the brain processes information while a person is watching a video. A major goal is to use machine learning algorithms to help understand the brain, and then to use what we understand about the brain to advance artificial intelligence.
So this is all I did. I wrote only 24 lines of code. So you can see a bit of a knowledge of NumPy, where I'm reading my medical data into a NumPy arrays, bit of a knowledge of a SciPy. What I got, this is the original PyQt graph example. I injected this code with those 24 lines of code, and Merry Christmas, I got a 3D data viewer running in no time, which could display most of the medical images, which was very useful for slicing the multi-dimensional images at an arbitrary angle. It is honestly a great tool for a CT and MRI, and it's very interactive tool. Let me show you the tool itself. Sure. Oh, it's coming? Yep, that's fine. We can't see on our screen, so I have to see it there. So, hang on, where is my mouse? Drag it to the right. No, that's fine, that's fine. So, the first, I can easily change the contrast level. I can see where most of the intensities are lying. I can go through the slices using the fast and the slow up and arrow keys, and this is what the researchers was looking. I can see the data quickly, and I can cut it at any particular arbitrary angle. I can zoom in, zoom out, very responsive. I haven't seen anything in Python so responsive. But before you load the data into the viewer, it comes up in a one single folder of 100,000 files, complete mess. You have to sort it in a way so that you can navigate nicely, and that's where PyDICOM comes in, which made our lives easy. And now Simon will explain to you how we use PyDICOM in our work.
That's right. I'll show you a great Python tool we discovered to process medical images. These are the raw medical images coming from the scanner, and here is where we have some flexibility to use Python for image processing. After this, we use other specialized packages that take care of outputting these beautiful maps depending on the type of information you want to extract from a brain scan. We will show you how we use Python in the processing stage of this pipeline. This is what you usually get from the scanner, a dataset with disorganized files dumped into a single folder. We use PyDICOM to output a nice folder structure that makes sense to scientists, that is, data sorted by patient name, study date, and the type of scan acquired within that study. As Gagan mentioned, DICOM alone is an extensive field that could easily take up a whole presentation. What you basically need to know is that DICOMs were created with the aim to standardize medical images coming from different sources. And this is done by embedding critical information into the tags. These are the headers Gagan was talking about. And we need to access these tags to create organized folder structures. With PyDICOM, you can navigate these structures and store them in handy Python variables, as I will show you in the next slide. I know it might seem like a mouthful of code. We have Python on top. We have Bash on the bottom. They achieve the same result. But tell me, which method do you find more intuitive? You're probably Python biased, so I know the answer for that. But for us, we find it very convenient as you read the headers and they become variables straight away. And there's less fiddling around with regular expressions. This is how we do it on the command line. Yep. So we start by importing the PyDICOM library. Now we assign the DICOM image to a variable. And now we can use the read function to access all of the headers. And here I can see all of the headers stored in a Python data structure. Even the pixel data is referenced here. I can also print, for example, patient name, or name, study date, and the place where it was acquired.
We're going to jump right in, and I'm going to describe work that we do in one of a couple model systems we work in. So in trying to understand the brain, we don't do so much in humans, though I used to. There's really a lot of reasons that model systems like animals are advantageous for answering neuroscientific questions. And one animal that we do a lot of work in is the larval zebrafish, which is probably not something that a lot of people here are familiar with or have seen necessarily. Maybe you have. So what's really cool about these animals is that when they're in the larva form, they're transparent, which means we can use special kinds of microscopes, in particular something called light sheet imaging, to basically sort of see through the animal and record neural activity from almost the entire brain at reasonably fast resolution and generate movies that look like this. So what we're watching here in the flashing bright colors is activity across the brain of this animal. We're able to see neural activity with light because these animals are genetically engineered so that their neurons express proteins that fluoresce when they're active under the presence of a microscope. And what we're seeing here is this animal. So this is the front, and this is the middle, and this is the back. It's a little weird to look at, almost kind of alien looking. This is a visual stimulus that's being presented to the animal, so we're sort of interested in how these creatures behave, how they respond to stimuli. The animal's actually paralyzed, but we detect its intention to move by recording electrically from its tail, more or less, which is a little bit ghoulish, but this here is an indication of the animal's behavior. The size of that circle indicates how strong the fish is trying to swim. So what you'll see just watching the movie is that every time the stimulus comes on, he tries to swim. And then there's this pattern, kind of complex dynamic pattern of neural activity across the brain. So on the analytics side, what we're really trying to do is just make sense of these volumes, trying to understand how populations of neurons, networks of neurons, encode information, allow this animal to sort of see what it's looking at, and then make decisions based on that, or at least respond based on that. The kind of analyses we do generate maps kind of like this one. So what we're seeing here is a decomposition of the kind of movie you just saw, where we can try to identify populations of neurons, groups, or regions of the brain, indicated by different colors in the map on the left, that then have corresponding associated time courses as shown on the right. So this is one of many different kinds of exploratory analyses we do to really just try to understand and recover structure in these data. So this is an amazing system because of the ability to measure everything, which has really kind of changed the way we think about a lot of problems. But the complexity of the behavior that these animals are capable of is sort of limited. I mean, they swim in response to stimuli, but maybe that's sort of not that much that the larval zebrafish cares about in the world. We also do a lot of work in the mouse. The mouse is a fantastic system for, I think, answering questions that really start to get to the kinds of things that we do and the kinds of intelligences that we possess as humans. The mouse also happens to be a system where we have incredible abilities to record neural activity, manipulate neural activity, and sort of monitor it in a large variety of ways. So a couple of those technologies are things like two-photon imaging. This does not let you record the activity of the entire brain, so unfortunately the mouse brain is not transparent. There are people thinking about how to do that, but it's pretty hard. What we are able to do is record from small fractions of the brain simultaneously. We also spend a lot of time thinking about mouse whiskers, which is not something that's particularly intuitive maybe, but it is a really important sensory modality for these animals. And we can also measure electrical activity. So we do a lot looking at pretty simple behaviors in these animals. For example, there's a lot of interesting questions to be asked just about how a really simple stimulus is encoded by the brain. So we do things like take a mouse where all of her whiskers except for one have been removed, and then we can flick that whisker or have the mouse flick that whisker against a pole and ask questions like how different sensory variables being encoded by that whisker are represented by the brain. So for example, we might have a couple neurons like these two neurons here, and we're looking at fluorescence signals using a technology like the one I showed you in the fish. And for each one of these, we're seeing neurons that are related to different aspects of what happens when the whisker touches the pole. So this is kind of the nitty-gritty of sensory encoding, but it's also kind of where everything starts. For us, it's probably more about vision. For them, a lot of it is about their whiskers. We can now do this over pretty large regions of the brain. So this is showing a map where every circle represents a single neuron, and the size of the circle is based on how strongly that neuron is encoding one of these two variables that I just showed, these two properties of the whisker. So I'll just jump into showing a very, very quick demo of this. And I recorded a movie in case the demo doesn't work. Just happened a couple times entirely due to Wi-Fi problems. All right, so I'm going to show at least one way of launching Lightning. So it's a Node.js server, but we have a lot of different ways to deploy it. There's a public server that we make available for free. And all of this is open source and free. And there's also a standalone OS X app that we built. So that's what I'm going to open right here. So this is built using something called Electron, which is a really cool new library for building standalone cross-platform applications. And in particular, we're using the Electron Menu Bar, developed by Max Ogden, which is an awesome project that was super fun to use. So what I just did is I launched our server, and it's just running up here in the menu bar. And I can open it, which takes me to the server here. And what I'm going to do is just show you the visualizations. So Lightning comes preloaded with a bunch of visualizations. And the really cool thing and core thing is that each one of these visualizations is just a different NPM module. So if people are comfortable with sort of front-end development, you can make an NPM module, and each one of these visualizations is one. But you can have tons of them in the community. So it gives us a way of having this ecosystem of visualizations. On your Lightning server, you can import new ones just by going to import npm. And now that my server is running, I can interact with it directly from within Python. So for example, inside this notebook, I can just connect to it here. And then I'm going to make some random data. And I'm going to generate a visualization of my random data. And that's going to show up here. All right, and this is an interactive plot. And I can do things like brushing. I can even access the attributes of the things that I selected. And the important thing to understand is that I'm generating this. This is just the scatter plot that's down here. And I could make any of these visualizations. And if somebody makes a new visualization, as long as they publish it to NPM, then anybody could grab it into their server and then start using that visualization. And they could use it from any language. So we have client libraries. This was my movie, just in case. We have client libraries that are in already Python, Scala, R, and JavaScript. I'm working on a Julia one just because I thought it'd be fun to play around with Julia. And a lot of our idea behind Lightning is that if we solve this basic problem once of sort of developing a cool interactive visualization, then that should then be available to all of these different languages, as opposed to having different languages kind of reinvent the way to do things. I do not think this applies to all aspects of visualization for sure. These are really targeting the sort of almost like widget-like visualization. Like I want to render a leaflet map, or I want to do some kind of cool tabular thing. But I think it's hopefully a cool way to start thinking about building visualizations that can target multiple languages and be used across languages. I also think there's interesting discussions to be had about where and how can we adopt common standards. There are really cool projects like Vega to try to standardize the description of a visualization. We're definitely interested in adopting that and using it. But I hope that Lightning starts to provide an ecosystem for developing and using these custom visualizations. So those are a couple links to it, and I'd be happy to talk to everybody about it more. The very last thing I want to talk about is a very recent project. We really just did this in the last couple months, called Binder. So the problem that we were trying to face here, this is something Travis, I think, really nicely alluded to in his talk as well, is really fundamentally a problem of sort of reproducibility, and in particular, how to take computational analysis that we do, which at least for us, and I think a lot of people here, is now happening in Jupyter notebooks, and how do we basically make it really, really easy to have, say, a GitHub repo where you have a bunch of Jupyter notebooks and maybe some dependencies, and just turn that into an executable environment that now anybody can use. And that was a problem we set out to solve, and the way we did it was building a system that leverages both Docker and something called Kubernetes, which was a platform recently open sourced by Google to basically deploy and manage Docker containers across a cloud. So I'm going to, again, do this one as a demo, and I want to highlight here an absolutely incredible person in my group who's built so much of this, Andrew Osheroff, and also Kyle Kelly, who's doing a lot of really exciting stuff in this space. We were very inspired by something Kyle did called TempMB, Kyle and others in the Jupyter community, and we've really built off that. And I want to just quickly show you how it works, at least by way of example. All right, so this is the main binder website. And the way this works is that we just put in a GitHub repo. So I'll do binder project / example requirements. And then we specify a set of dependencies. So we're trying to really not invent a new dependency system. We're trying to use specifications that already exist. And we know that a bunch of people have GitHub repos where they already have a requirements.txt file, for example. So with binder, you can specify a requirements.txt file. We just, a week ago, added conda environment files. You can also just give us a Docker file, but that's pretty intense. And a lot of people don't like to use Docker, and I don't blame them. So I'll just show an example of requirements. You can also attach services. So you can say that I want to deploy this notebook and have people be able to run the notebook. In order to do that, they need to have Postgres. So we can just add a Postgres service or add something like Spark. It's not like a cluster or anything. I should say we're doing this as a public service that we're making available. So there are limits to how much compute we can just be giving away. We really believe it's important that this be a public service. So in this case, I'm gonna make it. So I just click make. And I'm running on the dev server, which means we have a feature that Matt Rocklin really wanted because Matt Rocklin didn't want to write Docker files because he didn't want to set up Docker on his machine. So this is actually building now a Docker image for this repo, and we get to watch it happen. We have the little Domino's pizza tracker over here. We ordered Domino's just to like see the UI while we were trying to put this together. Well, my Andrew had never ordered, I don't know, he'd never ordered Domino's, I guess. I've done it a lot. So what it's doing is it's essentially building a Docker container for you. Under the hood, what's going on is that when you made that request to take your repo and build it, we basically took your things you wanted, like the requirements.txt file, put it into a Docker file, built that Docker container, and then pre-deployed it across all of the nodes running the Kubernetes cluster. So that just finished. And now I want to show you, we give you this little badge that you can embed in your repo. And we already made one for this guy. So here is this repo, and here is the binder. Now, anyone who comes to your GitHub repo can click this button and be dropped into the environment that we just created. So I'm just going to open that up. We had some port issues on Cal Visitor that we just fixed about an hour ago. And this should now be working. So here I am in an environment that has the requirements that I specified. It would have had the conda environment if I had done it that way. And now this is totally executable, so I can go in and work with it and generate figures and do whatever we want. So this is something that we're really excited about putting together. We've had really cool use cases already, a lot in the sort of teaching domain. People want to put repos available to all their students. Matt Rocklin has an awesome set of a binder repo that he made for a bunch of Dask examples. We then made it so the computers didn't have as many cores, which I think threw him off a little bit. But we're working on that. We want to have at least like 2 to 4 cores. I think that's reasonable. And I know. Well, that was sort of a mistake. We didn't intend them to be all available.